Glycolysis is the process in which one glucose molecule is broken down to form 2-pyruvic acid, also called as pyruvate. The glycolysis process is a multi-step metabolic pathway that occurs in the cytoplasm of animal cells, plant cells, and the cells of microorganisms. At least 6 enzymes are used in the metabolic pathway. In glycolysis cycle, the first process is the conversion of glucose into glucose 6-phosphate through the process called phosphorylation, in which glucose is phosphorylated into glucose 6-phosphate with the help of enzyme hexokinase, in which ATP releases energy to form ADP. Followed by that, glucose 6-phosphate is isomerized into fructose 6-phosphate, also with the release of energy from ATP into ADP with the help of enzyme phosphoglucose isomerase. And after that, this fructose 6-phosphate is phosphorylated into fructose 1,6-biphosphate with the help of enzyme phosphofructokinase 1. And after that, this fructose 1,6-biphosphate forms dihydroxyacetone phosphate and glyceroldehyde 3-phosphate molecule with the help of enzyme aldolase. And here, this dihydroxyacetone phosphate can be converted into glyceroldehyde 3-phosphate with the help of enzyme triose phosphate isomerase through the process isomerization, in which this glyceroldehyde 3-phosphate will form 1,3-biphosphoglycerate molecule with the help of enzyme glyceroldehyde 3-phosphate dehydrogenase, and by forming 2-NADH. And after this 1,3-diphosphoglycerate undergoes substrate level phosphorylation to form 3-phosphoglycerate with the help of enzyme phosphoglyceromutase. And this 2-phosphoglycerate undergoes removal of water to form phosphoenol pyruvate with the help of enzyme enolase. And lastly, pyruvate is formed from phosphoenol pyruvate with the help of enzyme pyruvate kinase, also by reducing ADP into ATP with the removal of energy. Krebs cycle. It is often also called as citric acid cycle. The first step in a Krebs cycle is known as a condensation step, in which it combines the 2-carbon acetyl group with a 4-carbon oxaloacetate molecule to form a 6-carbon molecule of citrate. In the second step, the citrate loses one water molecule and gains another as citrate is converted into its isomer, isocitrate. In step 3, isocitrate is oxidized, producing a 5-carbon called as alpha-ketoglutarate together with a molecule of carbon dioxide and 2 electrons, which reduce NAD positive to NADH. This step is also regulated by a negative feedback from ATP and NADH and by a positive effect of ADP. Steps 3 and 4 are both oxidation and decarboxylation steps, which release electrons that reduces NAD positive to NADH and release carboxyl groups that form carbon dioxide molecules. Alpha-ketoglutarate is the product of step 3 and a succinyl group is the product of step 4. CoA binds the succinyl group to form succinyl-CoA. The enzyme that catalyzes step 4 is regulated by feedback inhibition of ATP, succinyl-CoA and NADH. A phosphate group is substituted for coenzyme A and a high energy bond is formed. This energy is used in substrate level phosphorylation during the conversion of succinyl group to succinate to form either guanine, triphosphate or ATP. There are 2 forms of the enzyme called isoenzymes for this step depending upon the type of animal tissue in which they are formed. Step 6 is a dehydration process that converts succinate into fumarate. Two hydrogen atoms are transferred to FAD, producing FADH2. The energy contained in the electrons of these atoms is insufficient to reduce NAD positive but adequate to reduce FAD. Water is added to fumarate during step 7 and malate is produced. The last step in the citric acid cycle or Krebs cycle regenerates oxaloacetate by oxidizing malate. Another molecule of NADH is produced at the end of this Krebs cycle. In the electron transport cycle, electrons move along a series of proteins to generate an expulsion type force to move hydrogen ions or protons across the mitochondrial membrane. The electrons begin their reactions in complex 1, continuing on to complex 2, travels to complex 3 and cytochrome C via complex 4. The complexes themselves are complex structured proteins embedded in the phospholipid membrane. They are combined with a metal ion such as iron to help with proton expulsion onto the intermembrane space as well as other functions. The complexes also undergo conformational changes to allow openings for the transmembrane movement of protons. The NADH now has 2 electrons passing them onto a more mobile molecule, ubiquinone, in the first protein complex, which is also known as complex 1. Complex 1, also known as NADH dehydrogenase, pumps 4 hydrogen ions from the matrix into the intermembrane space, establishing the proton gradient. In the next protein, complex 2 or succinate dehydrogenase, another electron carrier and coenzyme succinate is oxidized into fumarate, causing FAD, flavin adenine dinucleotide, to be reduced to FADH2. The transport molecule FADH2 is then reoxidized, donating electrons to Q, becoming QH2, while releasing another hydrogen ion into the cytosol. While complex 2 does not directly contribute to the proton gradient, it serves as another source of electrons. Complex 3 or cytochrome C reductase is where the Q cycle takes place. There is an interaction between Q and cytochromes, which are molecules composed of iron, to continue the transfer of electrons. During the Q cycle, the ubiquinol, or which is known as QH2, previously produced, donates electrons to ISP and cytochrome B, becoming ubiquinone. ISP and cytochrome B are proteins that are located in the matrix that then transfers the electron it received from ubiquinol to cytochrome C1. Cytochrome C1 then transfers it to cytochrome C, which moves the electrons to the last complex. The cytochromes then extend to complex 4 or cytochrome C oxidase. Electrons are transferred one at a time into the complex from cytochrome C. The electrons, in addition to hydrogen and oxygen, then react to form water in an irreversible reaction. This is the last complex that translocates 4 protons across the membrane to create the proton gradient that develops ATP at the